Good evening. Welcome, everybody, again from wide and far to this month's, to February's Meetings of Minds. It's great to have you back. I can see the numbers pouring in now as I speak. I hope you are all well, keeping well, feeling optimistic, enjoying your role at the front line of changing health all around the world. That task goes on, and we are ever appreciative of the efforts that you all apply to achieving that, to moving that initiative forward. Today, we are gathered with a particularly special guest, in my humble opinion, somebody who I have the real privilege of being able to meet quite regularly since I took up my position here at Zinzino, working within science, working within research, and exploring certain kinds of nutrient areas as we do, and trawling through the research papers, you start to become familiar with certain names that guarantee quality, high impact, that are going to always extend an understanding of an area. And one of those names is the name of the professor that we have joining us today, which is Angela Rizzo. For me, there was a real goosebump moment and a pinch yourself in that very first meeting. So when we had our strategic group together, and there was, some great people on that board, no matter what. But for me, this was that moment of really touching base and making contact with somebody who you know through their work, and you have such a real appreciation and a value for what they've changed with regards to thought and insight and understanding and knowledge in particular areas that relate directly towards those nutritional choices, to what's happened nutritionally to the world through dietary shifts and changes. And the quality of the science that we make reference to, the quality of the evidence that we rely on to inform what we do, directly shapes the change we can make. And so, without further ado from me, because I can't wait to hand over really, Professor Angela Rizzo, welcome to the February Meetings of Minds. Without further ado, I'm going to hand over to you. I'm going to encourage everybody, as I always do, but definitely today, and I'm ready and waiting already with my notebook. Have your notebook ready. Take notes. Maximize the next hour's worth of information. And without further ado, Angela, I'm going to hand over to you.
Thank you, Colin. Thank you, everybody. Thank you for the invitation. Good afternoon. And thank you for the kind words about me. It's a great pleasure to be here with all these people from the Zinzino group and from the health area, interested in the nutrition and nutrition values of the components of our integrators and our health related to nutrition. I'm going to share my screen now. Okay. Okay, can you see it?
Yep, we can see that, Angela.
Okay, so I can start talking. And this afternoon, we will go inside a little bit on the mechanism of action of polyunsaturated fatty acid. I'd like to call the fatty acid building blocks, very smart building blocks of our body. Many of all you know something about polyunsaturated fatty acid, but I want to give you some more information about the biochemical function that they have in our cells. Okay, we are thinking in the past, 40, 50, 70 years ago, that fats are the energy part of our nutrition. Energy source, one of the more important. We know we have to have about 30% fats in our diet to give energy in our body. But we start to discover very early that the polyunsaturated fatty acids are essential for our physiological function. They are a vitamin for our body. That means that we need them and we cannot build them. Among the big class of polyunsaturated fatty acids, there are two classes that are essential: the omega-6 polyunsaturated fatty acid and the omega-3 polyunsaturated fatty acid. We know that these are fats, and from a chemical point of view, I want to show you some chemistry, but not going in the details. They are the chains, alkylic chains. And if we think about trans fatty acid or saturated fatty acid that we know that are not good for our health, that increase cholesterol and increase cardiovascular diseases. We can see from these very easy pictures, this, there is a straight chain of carbons, and they form a very hard structures with a very high point of melting and fusion, high melting temperature. So in our body and at room temperature, they are hard fats. When we insert one double bond or two or more double bonds in this alkylic chain of the fatty acid, you can see here we insert an angle in the structure, and more double bonds we have in this structure, the more the structure of the chain, you know, become a curve, and so the melting point of this fatty acid is very low, and within our body, 37 degrees, and also at room temperature, they are really fluids. We have a lot of polyunsaturated fatty acid in our diet and in our body. The two precursors are the omega-6 and omega-3 linoleic acid with 2 double bonds, and alpha-linolenic acid with 3 double bonds. These are 2 different classes and are precursors of all the omega-3 fatty acid, the alpha-linolenic acid, and all the omega-6 fatty acid, the linoleic acid. You can see that we have other longer and more complex fatty acid, you know, with 3, 4, 5, and 6 double bonds, and the structure becomes very much complicated. So we take from the diet the 2 precursors, the linoleic acid for the omega-6 and the alpha-linolenic acid from the omega-3 fatty acid, and within the liver, we can transform this fatty acid in the more complicated, the more fancy, the more useful for our body fatty acid that has at least 20 to 22 carbon atoms. So we have to elongate, and you see here, there are enzymes that are called elongases. So they elongate the chain, and they have more double bonds, 3, 4, 5, or 6. So we also need to insert double bonds, and we do this with the enzymes that are called desaturases. The omega-6 class are transformed mainly in arachidonic acid, 20 carbons and 4 double bonds, and we know the omega-3 are transformed in EPA and DHA. The enzymes that these fatty acids use are the same, so the omega-3 and the omega-6 are synthesized by the same fatty acid. Now we try to understand if the activity and the ability of the human body to transform the shorter and simple linoleic and alpha-linolenic acid in the more complicated and more useful fatty acid can be modulated and can be different from one person to another person. And we discovered, I put on the slide also some indication for the literature if someone is interested to go further in these details. We discovered that our diet components are able to modulate the activity of these enzymes. You see here the hepatocyte, so the liver cells, in which these transformations are made from the essential fatty acid to longer fatty acid. And many components from the diet, such as vitamins, folate, B12, such as polyphenols, carbohydrate, vitamin A, retinoic acid, but also cholesterol and saturated and polyunsaturated fatty acid are able to modulate the enzyme activity. This means that our nutrition all together with all the components determines how many fatty acids we are able to synthesize in our body. As information, we can say that high fat, saturated, rich in cholesterol diet inhibit the transformation of the LA and linoleic and alpha-linolenic acid in EPA, DHA, and arachidonic acid, while carbohydrates, polyphenols, and high-protein diets try to increase the activity of those enzymes. More important are the effects related to omega-3 fatty acid. We can have a diet rich in vegetables, and so we get the precursor, the alpha-linoleic acid, or rich in fish or fish oil, so we can get the EPA and the DHA from the diet. So what is better? It is better to get the precursor and to transform it in the more longer fatty acid for our body, or it is better to take directly the more longer and more important fatty acid. We know that there is a great competition between linolenic acid and alpha-linolenic acid. So the omega-6 with the omega-3. And usually in the diet of the European diet, but also the American diet, there is a higher amount of linoleic acid in our diet. So who wins this competition? Usually the omega-6 wins the competition. So if we take alpha-linolenic acid, only a little amount will be transformed in EPA and DHA. In fact, if we mark in some way the alpha-linolenic acid, the precursors, and we put in the body of in our organism, we will see that most of this fatty acid is utilized for energy. Only less than 5% is converted to EPA and DHA. And also, every person is different from the other. Every population is different from the other, because we have this enzyme have a different genotype and haplotype in the genes for every population. I found this very interesting representation of the metabolism of fatty acid to help people to understand what I mean. These are the enzymes, the elongases that elongates, and the desaturases that put the double bonds. In particular, for the desaturases, and this one, the fatty acid desaturase 1, the main responsible to synthesize arachidonic acid, ARA, and EPA, we can think about this enzyme as a valve. Okay? If you can see a series of tubes, we start from the precursors, and during this tube, you know, during the metabolism, you see the tube become little and little, so the different molecules comes to these tubes, but need to have the valve to be open, to be transformed in EPA, arachidonic acid, and DHA. If the valve is a little closed, like in people who has in the DNA a particular gene or a particular type of fatty acid desaturase, a type of genome that related to our ancestral, okay, precursors. This valve is closed because during the Paleolithic, the humans get the very long fatty acid from the nature, from the food that was very rich in this fatty acid. So they don't need to synthesize. And these enzymes from the ancestral haplotype are not very active. So if this valve is closed, only the omega-6 come through. The omega-3 will not come through. So we will have a lot of arachidonic acid synthesized and a lot of molecules coming from the arachidonic acid. You can see here in red a measure of arachidonic on a eicosapentaenoic acid ratio. you can see is very high in people who has this ancestral genome. If we go to people who have a more modern genome, they have a very open valve. So both omega-3 and omega-6 come to the tubes, and both of them are metabolized, and all the metabolites are synthesized. And you can see here the ratio between arachidonic and eicosapentaenoic acid is lower compared to ancestral genotype. So this is only to say to you that every person can transform the precursor in a different way in the body, depending on the genome, okay, and the activity of the enzymes. Okay, when we take these fatty acids from the diet and we transform in longer fatty acid, or we take the longer fatty acid from the fish food or from the animal food, they are integrated in our body. The main storage of the fatty acid in our body are the cell membrane, the wall of our cells. Here you can see depicted all the main mechanism, okay, of action of fatty acid in our body. We will have not time today to go through all the mechanism, and I will try to show you only these three mechanisms of action of fatty acid. So we take the fatty acid from the diet, we transform in our body, and we inserted mainly in the cell membrane. In particular, the polyunsaturated fatty acid. You can see here the structure of a cell membrane. The cell membrane is composed by a lipid bilayer. These lipids are called phospholipids. Each phospholipid has two fatty acids. One of these two fatty acids usually is a polyunsaturated fatty acid. Can be a longer one or a shorter one. Can be an omega-3, an omega-6, or either an omega-9 fatty acid. Within this lipid bilayer that is fluid, really fluid, there are a lot of proteins moving within the bilayer. These proteins are very important for the function of the cells because they modulate the communication between the inside and the outside of the cells. They modulate the transport. You see here channels within this protein, so molecules coming in and coming out. They modulate the fate of the cells and the metabolism. The structure and the function of the proteins inserted in the lipid bilayer depend strictly on the lipid bilayer composition and on the amount of the polyunsaturated that I highlighted here with the red that are present within the lipid bilayer. The lipid bilayer property depends a lot on lipid composition, depends a lot on the type of fatty acid that is linked in the phospholipid. If the phospholipids contains polyunsaturated fatty acid, so with a high number of fatty acid, with the structure that we saw in the previous slides that is really curved, we will have a very fluid state of the membrane with proteins moving around very rapidly. If the membrane is rich in saturated on trans fatty acid, then we'll have a very ordered structures, and it will have a very hard, difficult structure and difficult to function very well. Moreover, the cholesterol content is really important because cholesterol acts as a modulator of membrane fluidity. Studying on the membrane structure, researchers discovered that within this structure is not, homogeneous within the cell surface, but there are some little nano structures, so very little, 10 to 100 nanometer diameter of this structure that are called lipid rafts. What are the lipid rafts? these are structure as represented here, you know, as a raft, very hard structure that move within on the lipid bilayer, okay, because they are very hard compared to the fluid state of the lipid bilayer. I show you now a very nice pictures taken with a microscope, atomic force microscopy. And you can see here the fluid membrane, and these are the raft, the very hard structure. These structures are very rich in cholesterol, very rich in glycolipids, and very rich in protein. You can see here like little mountains, little peaks on this structure, and they move on the cells, and they move this protein on the cells, and they decide the signal of the protein that they bring on the cells. In fact, we can have two different kinds of lipid rafts. They are planar, and in the planar raft, we found a protein that modulate signal transduction, so signals from outside to inside the cells, receptors, and we have caveolae, invaginated part of the protein, of the membrane, in which the lipid raft modulate the endocytosis, so the capacity of the cells to take something from the outside and bringing this inside the cells. Taking a very quick look to the proteins that are present in lipid rafts, we found very interesting proteins related, for instance, to immunofunction. They are very represented on immunocells like T cells or B cells. We found a receptor able to modulate cell proliferation like EGF receptor, but also receptor able to modulate metabolism like insulin receptor. So think about diabetes and this kind of diseases. And also there are pumps, okay? These are pumps very studied in cancer research that are able to take out from the cells drugs, xenobiotic, chemotherapy. So these are responsible for chemoresistance. Knowing about the lipid rafts, other study has demonstrated that raft and membrane structure and function is linked to different diseases. And these are mainly neurological diseases like Alzheimer and Parkinson's disease, autoimmune diseases and chronic inflammation, cancer, diabetes, and different kinds of infection, bacterial, viral, and other pathogens. It is possible to modulate lipid raft function and lipid raft composition to try to use a therapy, a lipid therapy, to modulate or to help preventing these diseases? Yes. A lot of studies has demonstrated that omega-3 fatty acid, for instance, DHA, can be utilized as a lipid drug that is inserted in cell membrane and is able to disrupt, to modulate lipid raft structure. You can see here, this is the lipid raft rich in saturated fatty acid. You see very straight chain. These are monounsaturated or polyunsaturated fatty acid. This is the DHA. When the phospholipid incorporated DHA in the raft, you can see here, the raft changes shape. And this changing shape can change the fate of the proteins within the raft, can change the function of the cells. For instance, is in immune cells or in cancer cells, can change the proliferation of the cells. So the function of our polyunsaturated fatty acid in the membrane is very important for membrane functionings, and they function as a block that modulate membrane fluidity and lipid raft function. But these blocks, the fatty acid blocks, are not only, as components of this wall that can modulate the wall structures. So if the wall is more fluid or more rigid, but they wall, the phospholipids within the lipid layer, function also as a storage of polyunsaturated fatty acid. In fact, we go to the second part of this talk. The polyunsaturated fatty acids that are stored within the cell membrane, when a special signal touch the cells, stimulate the cells, can be released from the cell membrane and utilized to synthesize new signal molecules, lipid signal molecules that are called in general fatty acid derived metabolites. So we start from the building blocks of the membrane, the phospholipid, and with the signal that indicate, for instance, an infection and damage to the tissue, inflammation, and many other proliferation signals. There is an enzyme that is activated. This enzyme is called phospholipase A2 because it works on phospholipids and detach the fatty acid in position 2 and release the polyunsaturated fatty acid present on the building blocks on the membrane. The polyunsaturated fatty acid, either the omega-6 or the omega-3, are generating signals within or outside the cells that modulate metabolism, like lipolysis, synthesis of fatty acid and triglyceride, fatty acid oxidation, immune cell function and action, immune response and cell survival, and modulate pro and anti-inflammatory inflammation of the tissue or all the cells. So we go now a little bit more inside the inflammation process. The inflammation process is a good process for our body because let the cells respond to a problem that is present in this tissue, you know. The inflammatory activity starts with an acute inflammation. In few seconds after an insult to a tissue or to a cells, there is a release of many mediators. These are lipid or protein mediators that indicate acute inflammation. And as you can see here, in seconds and in minutes, we have an answer to this acute inflammation, a response. The first response is edema. This response is followed by a more strong inflammation that recall leukocyte to the place of the inflammation. You can see here polymorphonucleate infiltration. And this is also a very important phase of the inflammation because the leukocytes are trying to act against the problem that has caused the inflammation. And if everything goes well, within hours or days, we have to solve the problem and to go to the final phase of the inflammation that is called resolution of the inflammation. If everything goes well, we start from acute inflammation and we stop the process with the resolution of inflammation. The problem is solved. We don't have any problem in the tissue. If the resolution of inflammation delay and inflammation is prolonged in our body, we can have a problem of long-lasting inflammation that we know is connected with many diseases, and now we know for the COVID diseases is one of the major problems, the long-lasting inflammation in the body. Now, all these phases of inflammation are related to specific lipid mediators that are synthesized from the polyunsaturated fatty acid, from our building blocks present on the membrane, that indicates to the cells and modulate the inflammatory phase. You can see here, this is still the membrane, okay, of the cells. This is the phospholipase A2, that starts the process cutting the phospholipids and to have the polyunsaturated fatty acid free to be transformed in lipid mediators. If we use corticosteroids, for instance, to stop inflammation, the corticosteroids act, okay, on this enzyme. They inhibit phospholipase A2, and we stop the inflammation. You can see here in red the main omega-6 fatty acid, linoleic acid, here arachidonic acid, and in green the omega-3 fatty acid, alpha-linolenic acid, EPA, DPA, and DHA, the longer one. Each one can generate a different set of inflammation or inflammatory lipid mediators. These lipid mediators are synthesized from enzymes like cyclooxygenases, COX, lipooxygenases, LOX. This enzyme we know very well. This enzyme can be inhibited from aspirin-like drugs, non-steroids, anti-inflammatory drugs, but there are another class of enzymes that are not inhibited from non-steroids drugs, are not inhibited from aspirin-like drugs that are called CYP, cytochromal P450 enzymes. CYP, sorry for the English. So a lot of enzyme, a lot of lipids, we can have thousands of molecules that are synthesized for our smart building blocks. We will not go in details describing all the molecules because it's not of interest. But I want to show something, you know. In the acute phase, in the reactive phase, we have mainly, okay, the synthesis of what are called lipoxygens, because from lipids we insert in the molecule oxygen, so that's why they are called lipoxygen. Eicosanoids are some of them. There are many others that derives mainly from the red fatty acid, from the omega-6 fatty acid. And these lipoxygens mediate the initiation of inflammation. And if we have a very high concentration for a longer time, we go to chronic inflammation that is not good for the health. In the phase of resolution, we are mainly lipids mediators synthesized from the omega-3 fatty acid, EPA, DPA, DHA, and these mediate the resolution of the inflammation, okay? They stop the acute inflammation and restore the physiology of the system, of the cell, and of the tissue. Some of these lipid mediators are called with a name that indicates their function. They are called resolvins, indicating that they resolve the inflammation. They are called protectins, indicating that they protect against inflammation. They protect the tissue. Resolvins are mainly synthesized from EPA. Protectins are mainly synthesized from DHA. So the omega-3 derived lipid mediators have two main functions. They are anti-inflammatory agents, mainly the prostaglandins, the eicosanoids, okay? They act on white blood cells, on lymphocytes and macrophages, reducing, you can see here, the different cytokines that are responsible of inflammation, reducing the adhesion molecules that recall immune cells and that damage the endothelium. This is the first phase, the first function of these cells. But the new molecules that we discovered 20 years ago, and some of them we still don't know their function, are pro-resolving agents. So the anti-inflammatory agents stop inflammation. The pro-resolving agents repair, restore the tissue homeostasis, the resolvin and the protectin molecules. So you can see here, the red one from the omega-6, they mediate the initiation of inflammation, and the the green one from the omega-3 to resolve the inflammation. How much we synthesize from the omega-6 on the omega-3 depends on the composition of our membrane. So depends on the composition of our building blocks, the phospholipids, and depend from the lipid intake and from our metabolism within the cells. I will skip or say something about these lipid mediators. One of the first discovered is synthesized in the brain. Okay, it was discovered in the brain. That's why it's called neuroprotectin D1, because protect our brains, our neuron against apoptosis and necrosis. So it's very important for degenerative brain diseases. Going further to the activity of our building blocks of our fatty acid, they can be transformed in lipid mediators, but they can act directly as fatty acid, DHA, EPA, arachidonic acid. They can act on the proteins that are present in the cells, on the receptor, specific receptor that are present on the cells. They are called GPCR receptor, G-coupled protein receptors. This G-coupled protein receptor that we are discovering, like GPCR 120, is a receptor, for instance, for DHA. So it recognizes DHA that is released from the membrane that is present in our body, and it is stimulated from this fatty acid. We have this receptor in different kinds of cells, for instance, in the immune system, in the macrophage, okay, and the stimulation of GPCR 120s and the stimulation of inflammatory cytokine mediate the macrophage function. Moreover, it is present also in other tissue, like adipocyte, where it can mediate, for instance, glucose uptake, insulin function, so it can be responsible of lipid and glucose restoration during insulin resistance phase of diabetes mellitus. Not only the fatty acid recognize receptor on the cell membrane, but they also recognize receptors within the cell, within the nuclei, receptors that mediate many processes within the nuclei, and they mediate synthesis of proteins that again work on inflammation, that work on metabolism, that work on cardiomyocyte, for instance, function. So through receptor binding, we can modulate different metabolism in our body, for instance, glycolysis, lipogenesis, and fatty acid oxidation and inflammation. So through this mechanism, some of the mechanisms that I showed you today, omega-3 fatty acid play a major role in and function in our body. Some of this function that you see here, blood clotting, digestion, fertility, cell division, growth, are really demonstrated and are related to the amount of omega-3 fatty acid we have in our diet and our body. Some of these effects are still on the buds. I show you here in blue, you find the effect that are really demonstrated, and it's present with the, we can see dose correlation to the amount of omega-3 we take from the diet, to the amount of polyunsaturated fatty acid we take from the diet, like the reduction of triglyceride in our body. This is well demonstrated. Other effects, the red one, are relevant only if we take a very high intake of omega-3 fatty acid, more than 4 grams per day of omega-3, EPA, and DHA. For instance, you modulate coagulation and thrombosis. Some of these effects in humans are still under debate, like the cardiac effect on the myocardial efficiency on the ionic transmission, on the vascular system, because it's difficult in clinical study to find a dose-response relationship. Why? Because we need to have omega-3 balance in our body, but we need to measure this balance, because everybody is different from another person. I showed you before, because of the genetic enzymes, because of the different diet components that we take together with the fatty acid. So we need to measure the balance. We need to measure the balance. Because, as I showed you before, sorry, it's the repetition, we have functional variants of enzymes, we have epigenetic chains, so different population, inter-individual and variation in synthesis of very long chain fatty acid, inter-individual levels of PUFA omega-3 and omega-6, and so inter-individual levels also of lipid mediators that we generate from this fatty acid. This is a very interesting study showing that if we do experimental preclinical studies in animals, in which the animals are all the same, all with the same genetic patterns, all with the same diet, we found very nice results using omega-3 fatty acid treatment in preventing and in some cases in helping to resolve a lot of diseases. Okay, so we have very nice data. If we do in human intervention trial, a lot of the time, the high variability of the response, the dietary composition, make the very high variability in answer in clinical data we have, with contrasting data on the effect of the fatty acid. So I want to finish early, so to leave time for the questions. We have to measure the balance within the membrane of our body, within the cell that we are interested in. And we discovered many years ago, not me, but a lot of laboratory, that the erythrocyte membrane is a good model of how the building blocks are present in our body. There is a kind of relationship between the membrane composition of the erythrocyte and the membrane composition of the tissue, that be adipose tissue, liver tissue, and other tissue. We cannot take adipose tissue, liver tissue, or muscle and analyze. So we prefer to analyze the membrane of the erythrocyte. This is good, but it takes a lot of time. You have to draw the blood, to centrifuge, and to isolate the membrane and analyze the fatty acid. And moreover, you need to have fresh blood. So many years ago, we demonstrated that if you take some whole blood without isolating the red blood cells and measure the level of polyunsaturated fatty acid, arachidonic and EPAs, there is a straight correlation with what there is in the cell membrane. And this is very good, because we discover we do not need to have fresh blood, we do not need to have isolation of the membrane. And starting from this point, we starting collaborating with Ola Eide, that you know, are Bioactive Foods, developing the test that now Zinzino is using to measure the fatty acid balance within our body. Preliminary data indicate in the Italian population, for instance, that we have a ratio of omega-6 and omega-3 very quite high, 16 for arachidonic EPA acid, around 8 for omega-6 to omega-3, and that pathologists, many pathological problems increase this level of omega-6 in the body. In particular, as you can see here, inflammatory disease, very high level, cancer disease, metabolic disease, cardiovascular disease, allergic, cutaneous disease, and also central nervous system degenerative disease. This is the level that we measure in people without any diseases. So, to make the story short, polyunsaturated fatty acid, in particular omega-3, EPA, and DHA, are inserted in our building blocks in the cell membrane, where they modulate membrane fluidity, where they modulate raft and caveolae function, so the response of the membrane to different insult to different signals that comes outside from the cells. They regulate the proteins. They link to receptor outside the cell, within the cells. They are very smart, and when there is an activation of the enzyme, they are released within the cells, and they are transformed in lipid mediators that can be used to modulate and to resolve the inflammation. I skip this. Finally, now we know very well that we are what we eat, and this is particularly important for lipids. For the polyunsaturated fatty acid, as we described today, in particular, very nice effect of the omega-3 polyunsaturated fatty acid. Also, the monounsaturated fatty acid can have some positive effect on the inflammation, but there are other lipids that act with a similar mechanism. These are sphingolipids that we can take from the diet. These are short chain fatty acid that are synthesized by our gut microbiota, and these are the omega-6, the saturated and trans fatty acid, and the cholesterol that are very negative effect, okay, on our body in different tissue. You see here, brain, heart, liver, and more important, adipose tissue and the gut. So thank you for the attention, and I open to questions.
Superb. Thank you very much, Angela. Thank you. That was phenomenal. What an insight. Really getting under the hood and looking quite, impartially, separating and getting right to the science of the matter. There's been a question, there's been 3 or 4 people asked this. Will a recording be available? Yes, it will. And this is exactly why I said at the start, grab a pad and a pen to make notes. But yes, a recording will be available, and I totally understand it, because there is so much of this that underpins the primary message of really what a lot of people at Zinzino are about and the change that they're trying to make. Well, we have some questions, Angela,
yes,
have came in through the Q&A box, so I'll work through those now. I'm just going to, because we've got a little bit of time, I'm just going to go through them in order, and feel free to give your honest answer, or if there's something that you feel that you can't comment on, feel free to do so. So the first question that came in, funnily enough, was from somebody saying that they work with children who are dealing with autism, on the autism spectrum, and high arachidonic acid is a common finding. Do you have any comment on why that might be?
Yes. First of all, it depends from nutrition of the children, okay? Because usually children with autism of ADHD also, is common. They have a very, diet rich in carbohydrates, rich in saturated fats and omega-6 fatty acid. Secondly, usually the children has a higher level of omega-6 compared to omega-3. And this is related to enzyme activity in the liver that is not mature enough, and is also related to the development of the body. So we know now it's very clear that there is a very high omega-6 compared to the omega-3 in the autosomal disorders. And it's very clear that the omega-3 integration can help, not cure, can help in restoring the balance within the autism spectrum disorder. We did a study in the children, actually, in the young men, it was 16, 17, and we have a very good relationship between the omega-3 integration and the decrease of the symptoms of the autism spectrum disorders.
And that's really powerful, isn't it? I'm really glad you've emphasized that, Angela, that it can improve, not cure.
No.
And I think often, not just within that context, I think often what people don't fully appreciate is that when you can measurably change, alter, reduce symptoms, that in itself is powerful. That in itself is a tangible, measurable, positive improvement. I think all too often people are drawn towards kind of, really black and white solutions. Can it fix it or can't it? And actually improvement is the key, isn't it? Thank you very much. Right then, here's another one for you. which are better biomarkers for inflammation and cardiovascular risk? Is it measuring cholesterol, LDL and HDL, and homocysteine, or measuring the omega-6-3 ratio and the omega-3 index?
Okay, now we go to a very hot topic, it's cardiovascular. From the International Society, the major risk recognized is cholesterol and LDL level and homocysteine, like primary, association with cardiovascular diseases. There is no consensus on the level of omega-6, omega-3 and cardiovascular diseases. It is known that mainly there is a high saturated, high omega-6 in people undergoing cardiovascular problem, but there is no consensus about the useful of integrating omega-3. So I think you have to do both measures.
Mm-hmm. And on that note, I'm going to add a question because this crops up a lot, and I'm aware of the literature, but I think it'd be more powerful coming from you with regards to treatment. So when somebody has undergone a heart attack, is dealing with cardiovascular disease, and then we start to look at the role that omega-3 has in that situation. Do you have any comment on that with regards to the research is at?
Okay, there are different clinical studies contrasting, they don't measure the balance. People have different diets, different genetics. The more important study is VITAL with a very high amount of EPA taken together with statins. So a synergistic effect of omega-3 and classical statins drugs. In this case, there was a reduction of risk of heart attack in people taking both of the drugs. Okay.
So again, more combined rather than trying to isolate one solution. Yeah.
Yes. We are talking, about risk prevention, of the disease, and in people that has a lot of different disease factors.
Mm. Can we stay with disease? I got a lot of questions, and normally, Angela, I steer people away from these questions, but I know that this is an area you're confident with. So if you don't mind, I'm going to stay with disease for a moment. And
if I can answer, I tell the truth what I know.
Yeah, and that's all we ever ask. And this one, because this is another one that crops up a lot, so it'd be interesting to hear your take. But if you're not comfortable answering, please say. What is the role of omega-3 in treating Parkinson's disease, and how omega-3 could potentially help with Parkinson's disease? What are your thoughts on that?
Okay, I'm not a neurologist, so I'm not very fan of this. While I studied a lot of cardiovascular, I'm not a neurologist. Also there, in Parkinson and Alzheimer, there is a lot of oxidative stress and in the brain, and omega-3 can prevent the inflammation and the oxidative stress that is present in Parkinson and Alzheimer's disease. But there is a quite correlation on what is missed, low omega-3, and the incidence of the diseases. There are not a lot of study on treating the disease and getting the results. Mm. I don't know if I explain it well. We know that in Parkinson, in Alzheimer's disease, there is a low level of omega-3 in the body, and also from autoptic tissue, also in the brain. Mm. Actually, we don't know if this is the cause of the disease or con-cause of the disease. And there are not enough robust trials that indicate that omega-3 can cure or help in these diseases. Yeah. And mainly they have an anti-inflammatory, antioxidant effect in the brain.
Hmm. I think that just on that, because this is something that crops up a lot, I think with regards to the more holistic, preventable and preventative strategy, that making sure that we have a sufficient index and ratio, it shouldn't really be targeted towards the notion of one condition, and we should be looking at kind of optimizing profiles with regards to how they should be as a preventative strategy, and not trying to target individual ones.
And if you have a gene that drive you to the Alzheimer's disease, you cannot with omega-3 change your genome, and you can try to have a better disease, a better life, but the disease is there, so you're looking for another therapy.
Yeah, absolutely. We've got time, if you're happy, we've got time for one more question.
Yes.
And this is an interesting one. There's loads of questions, and I apologize, people at home. I always try to get through as many questions as I can. So if I haven't chosen yours, I'm sorry. I'm trying to go through and get the ones that I think really the most relevant. But this is an interesting one for us to finish with, I think. Is there a downside to taking more balance oil than strictly necessary? Is it possible for the membrane to become too fluid, for example?
Okay, we know that the European Safety Authority say to us that we do not have to take more than 5 grams per day, okay, of omega-3, because we go on coagulation problems. This is the first thing. The second thing, there is a kind of protection of our membrane. So if we have too much polyunsaturated fatty acid, there is a lot of cholesterol being synthesized to counteract, okay? Mm-hmm. So I think to stay in the allowed dose and to test for the balance, because 2 grams will be enough for you, 1 gram can be enough for me. So the balance will give us the answer for the amount to take. No go under 5 grams a day, because we know we will have a coagulation problem. If we need to have a surgery or whatever, we will be in trouble.
Mm. Superb. Like a textbook answer, Angela. Yeah, and just to add to that, because this again is a question that crops up a lot, and that's why we have the test. And that's why in science and in research, we rely so much on the capacity, the accuracy of our tests, so that it can inform decision making. And that's what the test is about for you. This is exactly what it's about.
And if I can say some word about the results of the test, we are studying now in our group, you will know, we discussed with Ola many times. The test says that the balanced oil with the polyphenols has a different effect of the normal omega-3 integrator that does not have polyphenols. And this is related to the enzyme activity that I showed you before. So the effect directly of the oil composition. Well, we will go further in this detail when we will analyze better our big database on this test.
Yeah. Which everyone, believe me, the amount of requests we get for that analysis is building day by day. Mm-hmm. Listen, thank you, Angela. That was just incredible. I knew it would be. I felt like I was back at university, just pen out, taking loads of notes. It was incredible. And I know and I hope that everybody at home benefited from that as well. The recording will be made available at another time, in your own time. Go through it again, press pause, take notes. We can never learn enough. We can never learn too much. And it's always good. And I think what you've really brought to this, Angela, this afternoon is that real, authentic, balanced approach where you are counterbalancing evidence and looking at things in a really grounded way. And that's what we're about. We don't need to invent things and make things up, because the science is there and that's compelling enough. So Angela, on behalf of everybody at Zinzino, thank you so much for your time, expertise and wisdom. And to all of you who've joined us again, thank you for taking time.
Can I say some words? Can I say some words? I take the occasion that a lot of people from Europe is here. You see on my back, this is the picture of our dome in Milan. This picture was taken 2 years before ago. When we started our lockdown, 1st of March, nobody there. So Milan is open now. Italy is open now. Come back to Italy because it's a wonderful country.
Love to. I'm going to take that as a personal invite. So I'll start booking my flights, Angela. I'm on my way.
A little advertising, I go. Okay.
Thank you.
Bye to everybody. It was very interesting.
Thank you all. Thank you for being a part of changing health on a global scale. It all comes down to you. Taking this information and making it matter and embedding it within your communities. That means so much to all of us here, knowing that the work we do reaches those people where it can make the biggest difference. There is no meetings of minds in March because we have director's trip and there is a ton of things that are going on in the preparation and delivery of that. So we will be back in April, same time, same place, date to be confirmed. And until then, stay well, keep doing what you're doing. Thank you again, Angela. Everybody stay well, and we look forward to hosting you all again very, very soon. Bye now.